going to demonstrate Lab 2, where I will use restriction enzymes to digest 2 plasmids. Plasmids are small, circular pieces of DNA. Each of the plasmids I will be using have particular genes of interest. Restriction enzymes can cut the plasmids at specific locations, allowing us to separate out those genes from the rest of the plasmid. I will use the cut pieces of DNA to create a new plasmid in Lab 3. Here are the reagents and equipment I will use to conduct Lab 2. I have tubes with each of the plasmids. pKAN-R or K, pARA or A, a tube with a mix of 2 restriction enzymes, BAMH1 and Hindi3, a tube of 2.5x buffer, and a tube of distilled water. First, I will combine each of the plasmids with the enzymes and buffer and use a centrifuge to make sure everything is mixed. Then the samples will digest in the water bath for 1 to 2 hours. I will also do a control setup. The control tubes will have the plasmids and buffer, but instead of restriction enzymes, I will use distilled water. To set up the experiment, I need to label the tubes. K+ and A+ are for the experimental setups. K- and A- are for the control setups. Now I'm ready to add the reagents. First, I'll add 4 microliters of buffer to each tube. Then I'll add 4 microliters of K to the K+ and K- tubes. I'll then add 4 microliters of A to the A+ and A- tubes. I'll add 2 microliters of restriction enzymes to the experimental setups, K+ and A+. Finally, I'll add 2 microliters of distilled water to the control setups, K- and A-. Now I'll centrifuge the tubes. Notice I'm evenly spacing the tubes to make sure the centrifuge is balanced. I'll run the centrifuge for a few seconds to make sure everything is mixed and everything is at the bottom of the tubes. Now I can transfer everything to a floating rack and then into the warm water bath. Only the bottom of the tubes, where the reagents are, need to be under the water level. The plasmids will digest in the warm water bath for 1 to 2 hours. I can leave the digested samples at room temperature, but if I am not moving on to the next lab right away, I can store them in a refrigerator or a freezer. In the next lab, I will use the digested samples to create a new plasmid.